Okay, this is viruses part 2. I was called away for a meeting just then. So pick up where I left off on slide 4, looking at the general characteristics of a virus and the bits and pieces that make a virus up. So we have a virion. A virion is the entire virus particle. The nucleic acid plus the protein coat plus the envelope, if a virus has an envelope. And the virion would also include things like spikes. The protein coat is the capsid, and then the nucleic acid material gets packaged up inside the capsid. Also inside the capsid you find enzymes and other chemicals that the virus will use once it's infected the next host cell. The capsid is comprised of little subunit pieces. The capsid is made up of proteins. These subunit proteins are called capsomeres. So each one of these little dots here is a capsomere making up the capsid. The capsid plus the nucleic acid is called a nucleocapsid. For a non-enveloped virus or a naked virus, the nucleocapsid is the same thing as the virion. For an enveloped virus, you need the nucleocapsid plus the lipid envelope, and that makes up your virion. Spikes, we'll look at spikes in more detail in a second, but spikes are how the virus recognizes and attaches to the cells. They are protrusions, again typically ending with proteins. These little red dots on the end here are proteins. Proteins because viruses work by shape matching. And I've talked a lot about the idea that proteins have shapes, and there's something on the surface of our cells have all kinds of stuff on them, and our viral spike typically fits into a receptor that's on the cell surface. On to slide 5. The difference between enveloped viruses and non-enveloped viruses. Enveloped viruses have a lipid bilayer envelope around them. So there's the genetic material inside the capsid, and then on the outside surrounding the capsid is another layer, and it's typically made of the same stuff that that cell membranes are made out of. And in fact, you can see here through the process of budding, you can see, and we'll look at in more detail later on, the idea that the virus pushes itself out through the membrane of its host cell and picks up the envelope as a result. Non-enveloped viruses don't have the envelope, obviously. They are also known as naked viruses. Typically, the presence or absence of an envelope is very important in how the virus gets in to a cell and also how the virus gets out of the cell. It's also kind of important in terms of how resistant the virus is to disinfectants. A naked virus doesn't have the envelope. The envelope is kind of an easy target, and simply the non-enveloped virus doesn't have that target for us to attack. On to slide 6, virus shapes, and in the textbook on page 335 there are virus shapes. Icosahedrals, these are a shape made up of triangles, 20 equilateral triangle faces. We have helical capsids. We have our viral genome here, and then we have the capsomeres built around and round and round, making these kind of rods of protein. And we have these complex shapes. This shape down the bottom here is the shape of a bacteriophage, and we will be talking about bacteriophages later on. Inside the capsid can be DNA or RNA, but never both. And this is one major method for classifying viruses, whether they are DNA viruses or RNA viruses, whether they have single-stranded or double-stranded. You can have single-stranded and double-stranded DNA viruses. You can have, mostly RNA viruses are going to be single-stranded, but you can have different varieties of what we call strandedness in our virus. We also have variation in the structure of the genome. Some viruses have linear genomes. Some viruses have multiple strands of linear genomes. Some viruses have circular genomes. So just know that there's a lot of variety in what a virus can be. Attachment, we'll take a look at attachment in more detail. Protein components for attachment. A phage, which is the short name for a bacteriophage, that has these tail fibers, and they work very similar to the spikes on animal viruses. They stick into receptor structures on the cells. Slide number 7. Slide number 7, sorry. Again, characteristics of viruses in the book on page 336 and over to 337. There's those tables containing many different kinds of viruses. Viruses being generally considered as not living things. They don't follow the same kinds of conventions, but there are families typically, and genuses and species of viruses beyond their classification based upon their nucleic acid, based upon things like their capsid, based upon things whether they have an envelope or not, based upon size, based upon things like the polarity of their genome, which we'll mention later on. Many of our virus families end in this term viridae or viridae, and there's no consistent pattern for naming viruses. So coronaviridae, from the structure of the virus, corona meaning crown. So I guess someone looking at the virus actually would be interesting to know when that term was coined and what they were looking at through a microscope, or maybe through an electron microscope, or maybe through some other kind of mechanism. Corona means crown, and that's where we get the coronavirus, or the coronaviridae. Other viruses are named for where you find them, or where they were first discovered, or where they first were seen. Bunyaviridae, for a virus that was found in Uganda, in Africa. Many of our genus names end in virus, such as enterovirus. Entero means within, or it means within the intestines. So an enterovirus is an intestinal virus, for example. Many of the species names are named for the disease. So the poliovirus causes poliomyelitis. And we often typically refer to viruses by their species name, such as poliovirus, or you can look at all the examples in the book there. Rabies, for example, and HIV. A lot of variation in the naming scheme. Okay, next on to slide 8. In addition to that variety in how we keep track of the names and classification of viruses, there are also some other ways that we can generalize about viruses. And when we generalize like this, we're typically talking about unrelated viruses that happen to do the same thing. So enteric viruses, enteric means the intestines. Typically they are transferred in the oral fecal route, and that's how they get out of one person and into the digestive tract of the next person or the next animal. Or we might have a respiratory virus. Again, there's a whole lot of different viruses that attack the respiratory system. It's kind of an easy target. So the virus gets in, makes you cough and sneeze. The virus particles are expelled into the environment, and then they're breathed in by the next host, and they get to where they want to be, and they're coughed and sneezed out again. So the route of infection, whether it's the oral fecal route or whether it's the respiratory route. Some other kinds of ideas about viruses. We can have zoonotic viruses. These are viruses that can be given to humans from other animals. And we have arboviruses from arthropod born. And these are viruses that are spread by arthropods. So we looked at the idea of arthropod vectors last time. There are many examples of viruses that are spread by arthropod vectors. Yellow fever, dengue fever, West Nile encephalitis, cross encephalitis. Sex-gene transmitted viruses too, an important type of virus. Okay, so last for this section, let's look at the virus that is on everybody's mind. COVID-19 is the name of the disease caused by the virus that was labeled SARS-CoV-2. SARS standing for Sudden Acute Respiratory Syndrome, and the CoV part for coronavirus. 2 because it's the second SARS-like virus that's been discovered. So what do we know about the coronavirus? We know that they are envelope viruses. We know that they are RNA viruses. And we'll look at what it means to be a positive sense or a negative sense virus later on. They are respiratory viruses. They travel from person to person through coughs and sneezes in particular. And our virus here has what we currently believe is a zoonotic origin. The original SARS virus 2 had a zoonotic origin. It was out there in animals, and then it was passed to humans. So if it's passed from animals to humans, we can describe that virus as zoonotic. Has this structure here. Here's our RNA in the yellow. The RNA is all curled up, and then it has a helical capsid built around the RNA, and then that nuclear capsid is folded up, and the whole thing is surrounded by a membrane envelope. So in the center here would be our nuclear capsid, the entire thing with the membrane surrounding it would be our virion. It's a relatively large virus, 120 nanometers, and it has a kind of a regular genome size of about 26 to 32 kilobases. That's 26,000 to 32,000 bases. And just for comparison, E. coli has 4.6 megabases, or 4,600 kilobases, or about 4.5 million. Humans have about 3 billion bases. That Pandora virus I mentioned earlier as being an unusually large virus has 2.5 million bases. So a base, that is our A's, T's, G's and C's. So you can read the human genome as A's, T's, G's and C's. 46 chromosomes, you would read all those letters, there'd be about 3 billion of them. You would read all the letters in an E. coli, there'd be around 4.6 million. You would read all the bases in our SARS-CoV-2, there'd be about 30,000. So why are viruses not organisms? Because they have this phase when they are entirely inert. When they're outside of cells, they certainly are not alive because there are no chemical processes occurring. But then when they get into viruses, into a host, they require a host. They are obligate. They have to get inside the host, and they steal all the host stuff. They either get inside, they steal the machines, and they steal the materials, or they get inside and they just steal materials. They also don't have a cellular structure. So even though they have some of the envelope viruses do have a lipid bilayer envelope, it's not quite the same as a true, say, cytoplasmic membrane or a viral envelope is not the same thing as a plasma membrane around a cell. And then many of our naked viruses don't have any kind of membrane. General sizes of a typical virus in the nanometers, tens of nanometers on the small side, many hundreds of nanometers on the large side, a wide range of sizes. Now, the parts of a virus, the virion, the capsid, the nucleocapsid and the spikes, know what the difference between an enveloped and a non-enveloped virus, know the different kinds of nucleic acid material you might find in your virus, whether it's DNA, whether it's single-stranded or double-stranded, whether it's RNA, whether it is the plus strand or the minus strand, the positive sense strand or the negative sense strand. You recognize those simple viral shapes. Know what the terms enteric, respiratory, zoonotic, and arbovirus mean as they're retroviruses. And I'll give you a task to look through the tables on page 336 and 337. Take a look at the nucleic acid type, the strandedness, the presence or absence of an envelope, and I think coronavirus is in there, COVID-19 in particular, but which kinds of viruses cause these diseases? COVID-19, measles, chickenpox, influenza, and AIDS.